It's exciting to see us actually being able to say, here's why you have kidney disease. And to me, that is really rewarding.
Great, I have a diagnosis, but what does that mean for me as a patient? And what does that mean for me as the caring physician?
It's so valuable for patients to have a name to their disorder and to know that this is why I have it and this is what's going to happen to me.
This is a really important step for helping us in the future to combat this disease.
Welcome to another episode of ASN Kidney Translation. I'm your host, Dr. Matthew Sparks from Duke University, and today we will review articles from the ASN portfolio of journals that each bring fresh insights into the role of genetics in kidney disease. First up, we'll review a meta-analysis from CJASN, examining the use of genetic testing for adults with kidney disease. This systematic review brings clarity to the question that nephrologists face daily. How valuable is genetic testing in routine CKD clinics? Next, we'll explore a study published in JASN focusing on a novel approach to systematically screen for the pathogenic MUC1 variant that is left off of traditional kidney gene panels. This genetic mutation is a known culprit of autosomal dominant tubulointerstitial kidney disease, and the study offers a new look at how precision medicine is advancing diagnostic tools in nephrology. Finally, we'll delve into a study from Kidney 360, which investigates the clinical, pathological, and genetic features of patients with digenic Alport syndrome. This work highlights how complex genetic landscapes influence disease progression and prognosis, particularly in conditions involving multiple genetic factors. Stay tuned as we unpack these important findings, bringing you insights on how they could impact patient care and the future of nephrology. Let's get started. I'm joined by Dr. Dervla Conington from Western University in Ontario, Canada. She is the Eugen Drouleau Chair for Kidney Research and Innovation at the Schulich School of Medicine and Dentistry. We'll discuss her recent paper in CJASN titled Utility of Genetic Testing in Adults with Chronic Kidney Disease, a Systematic Review and Meta-Analysis. Welcome to ASN Kidney Translation. We're happy to have you here.
Thank you very much for the invitation.
Well, this is a very timely paper. Genetics becoming more and more popular, but it's also facing the community with a lot of questions. What do you do with the results? How do you order it? How do you consent? How do you do genetic counseling? So let's just start at the basics. How did this project start for you?
That's a great question. We've always known that genetics plays some sort of role in chronic kidney disease, but in the last 5 to 10 years, we've really seen a rapid emergence of data showing a more significant role in disease causation, not only in childhood onset chronic kidney disease, but also in adult onset disease. But the major problem we were noticing in the data is it was particularly difficult to extract the exact role in adults as opposed to children. And when we're trying to devise guidelines on who needs testing, we really needed this data. So what we really wanted to do was decipher what is the exact role of genetic testing in adult nephrology practice. And when you're sitting, let's say, in a general nephrology clinic, how do you decide who needs testing?
Yeah, this is a really important question. Let's just think about it. So first, okay, yes, how many have a positive result? But two, what happens after that positive result? How does it affect the patient? How does it affect their care? Is that sort of where you wanted to get that granular with these studies?
Yes, we're looking studies from 2005 up to 2023, and really we wanted to answer 4 basic questions. What is the diagnostic yield after doing genetic testing in an adult with chronic kidney disease? And what I mean by that is how many of them have a confirmed molecular cause of genetic kidney disease? The second we wanted to ask is who needs this type of testing? The third question we wanted to explore was what is the optimal type of testing in these patients? And finally, with the data that was available, we wanted to determine what is the clinical impact of getting a genetic diagnosis. So great, you have the diagnosis, but how did this ultimately impact your management and patient level outcomes?
It sounds like a lot of lofty goals. So I guess let's go ahead and just talk about what were the methods that you and your group used to try to answer those 4 questions.
As the title suggests, this was a systematic review and meta-analysis where we screened over 7,000 manuscripts that performed genetic testing in an adult population with any type of chronic kidney disease, and it was from January 2005 to December 2023. We used this duplicate independent data extraction method, and ultimately we ended up with 60 studies, including over 10,000 adult patients. And by adult, we mean they were over the age of 18 at the time of genetic testing. And this is the methods we used to come up with our findings.
So there were 7,000 studies that had information about genetic testing.
Yes, there's a lot of data out there. We screened 7,000 manuscripts trying to extract data purely on an adult population. And this is where we whittled down to the number of 60 studies, including 10,000 adults.
So my guess is that these studies had to first be in adults. Two, you had to have at least the granular detail to talk about what happened after the diagnosis was ascertained. And so it had to be really well conducted and a group of patients, not just a single case report or something like that. Is that correct?
Yes, we included all studies that addressed an adult population. We had to be able to extract the methods used to perform the genetic testing, as well as the subtype of chronic kidney disease. We excluded case studies, case series, those that looked at single gene testing, so just one gene testing. You needed to have a more comprehensive approach to testing in a predominantly adult population, or in a population where you could extract the information on adults.
Where were these studies performed at? What countries principally produced the data for the meta analysis?
It was a worldwide population, and many studies, North America, Europe, Asia. We did include studies where we were able to access English only manuscripts, or we could avail of translation.
What was the findings in terms of diagnostic yield of genetic testing in these studies that you mentioned?
So our findings found a pooled diagnostic yield of 40% in adults who undergo genetic testing. And this means that 40% of the people in these studies got a genetic diagnosis. This varied a lot depending on what we call the presumed subtype of chronic kidney disease was pre-testing. It also varied by the patient characteristics. So let's say patients had what we now refer to as genetic risk factors, so be it a positive family history of chronic kidney disease, or perhaps they had what we call extrarenal features of disease. That's where you have chronic kidney disease, but you also have an additional organ affected, let's say the brain, the eyes, the ears. These patients had a higher risk of genetic kidney disease.
So that's an interesting point. And sometimes when we're thinking about genetic testing, and maybe we can just sort of pause right here and talk about this, is if a patient doesn't have these extrarenal symptoms or family history, should you still consider genetic testing in them?
Absolutely. Each patient needs to be taken within themselves. And as I said, there's certain risk factors for genetic kidney disease, but there's also certain subtypes of chronic kidney disease that warrant genetic testing. Additional study that we did within this was to look at what subtypes of chronic kidney disease, are you're most likely to establish an underlying genetic diagnosis. So certainly some types of chronic kidney disease, be it cystic kidney disease, ciliopathies, tubulointerstitial kidney disease. These diseases, if you suspect them pre-testing, you have a high probability of establishing a genetic diagnosis post-testing. But one really important feature of these studies was that some patients have what we call chronic kidney disease of unknown cause. This is where your standard diagnostic tools that we're currently using, be it artery markers, imaging, and even your kidney biopsy, despite undergoing all this testing, you do not know what the exact cause of chronic kidney disease is. And within our study, we found a high proportion of these patients, almost 20%, you could establish a genetic diagnosis after undergoing genetic testing. So it's very important to remember that when we're doing testing, not everybody's going to have a positive diagnosis. And what's important to remember, let's say if you're doing testing for a PE, you wouldn't expect 100% of people that you're testing for a PE to have a positive CTPA. That means you're probably not doing enough testing. So although a 40% diagnostic yield may seem low, if we're only screening people who definitely have genetic kidney disease, there's certainly other people that we're missing.
So what you're trying to say is that the 40% is a higher rate than what we probably would find in a general nephrology practice.
Yes, there's two major studies that contribute a lot of data to the adult population. It's the Emily Groupman paper and the Dahl paper. They predominantly report in a pediatric general population. And what we have done within this study is we have developed a sort of selectivity criteria. So we rated these 60 studies from 0 being the most unselect general chronic kidney disease population to more selective populations because they have an additional risk factor to genetic kidney disease, be it family history, be it the extrarenal features, to disease subtypes or CKD subtypes where there's a higher proportion that have a genetic contribution. So what we found was with a general chronic kidney disease population from our current study that the mean or median diagnostic yield is 17%. If you have extrarenal features of disease, again, the family history, the extrarenal features, this increases up to 38%. And then certain subtypes of chronic kidney disease, if you do genetic testing, as for example, tubulointerstitial kidney disease and certainly cystic kidney disease, the probability of establishing a definitive genetic diagnosis after testing often is higher than 50%.
Great. Well, it sounds like that, even in a general practice, this is a test that could help. But I think we talked about the diagnostic yield. We still have other questions that need to be answered from your study. So do you want to move on to your second question that you had?
So the second question we were trying to look at is who should have genetic testing? And again, this goes back to the selectivity criteria that we were trying to develop. So we strongly feel that anyone who has a positive family history based on these data have a higher probability of having a genetic diagnosis, and those with extrarenal features of disease have a high probability of a genetic diagnosis. However, that doesn't include patients who have negative family history because of recessive disease or de novo mutations. So it shouldn't be the sole criteria used for genetic testing. Interesting finding that we didn't potentially suspect was that there was no decline in the diagnostic yield with increasing age of testing. What we mean by that is that the older adult, we would have automatically assumed there's less genetic contribution and more, let's say, environmental or polygenic contribution. But both in our analysis and when we divided by subcategories, so we looked at those who had a CKD onset greater than or equal to 35 and end-stage kidney disease onset greater than or equal to 50, we didn't find any difference in the diagnostic yield after genetic testing.
One of the things that I've often seen is that the family history sometimes starts with the genetic testing. And kidney disease is a silent disease. Oftentimes, individuals don't know they have it. It has variable penetrance, these genetic mutations. So I agree, like looking at family history as your stop or start for genetic testing is very problematic in a disease like kidney disease because of those issues. It's been very enlightening to see that really changing how families are seeing the diseases they have because of genetic testing, I think has been a very positive movement in the field of nephrology. And I'm sure it's happening in other disciplines as well.
When we're developing guidelines, we do need to be cautious that we're not just using one criteria that isn't really rooted in the data that we currently have available. And the example of this is the age of onset of chronic kidney disease. We currently don't have the data. Now, one gap in the data is there isn't that many studies including patients above the age of 50. So one area we need to improve to ensure that we have equitable access to testing for all is to ensure that we actually study these patients. But for those that were included in our current studies, we didn't see a decline in diagnostic yield with increasing age. And this is reminiscent of what the current KDIGO guidelines are for genetic testing, suggesting that there shouldn't actually be an upper age limit for genetic testing in chronic kidney disease.
Figure 3 is particularly interesting to me. It shows this reclassification disease based upon what was known from the clinical and pathological findings, as you mentioned earlier, and how genetic testing changed this. Can you describe this figure and what exactly it is saying about how genetic testing really changed how the clinicians saw the disease?
Absolutely. This figure is a little bit complex and we like to use color in it. But what we really wanted to show here is that in people who get a confirmed genetic diagnosis, what was their diagnosis before testing, the so-called presumed diagnosis of chronic kidney disease? And when they underwent genetic testing, what was the diagnosis they got after testing? So take, for example, cystic kidney disease. When you have assumed diagnosis of cystic kidney disease and you undergo testing, a large proportion of those patients, we find a mutation in a gene that's known to cause cystic kidney disease. However, that's not the case in all cases. For example, in some patients who you suspect have cystic kidney disease, when you actually do the genetic testing, you find that these patients have a mutation in a subtype of chronic kidney disease that is entirely different. So, for example, some patients are diagnosed with tubulointerstitial kidney disease or ciliopathies, which is a very different diagnosis, both in terms of management, prognosis, surveillance, and ultimately information for the family. So we found overall in all subtypes of chronic kidney disease, there was a 17% reclassification rate. That means the disease you thought you had before genetic testing was in fact different. So that means when you're sitting in a general nephrology clinic, we can assume that potentially up to 1 in 5 patients who we think we know the diagnosis, we maybe don't. And this diagnosis can really only be established after you do genetic testing.
Well, I just want to reiterate that. So, first we had a 20 to 40% diagnostic yield, and then we have a 17% reclassification rate. And just make sure I'm clear about this. These are in patients that actually did have a positive finding on genetic testing.
Yes, we just looked in patients in whom we confirmed a genetic diagnosis, and we looked at the pre-testing diagnosis and the post-testing diagnosis and to see was there concordance. And in 17%, the pre-diagnosis was very different from the post-diagnosis.
Do you think there's any way that you can look at whether or not here's the diagnosis that you think they have, but the genetic testing was negative? Would that change the diagnosis at all? Did y'all look at that question?
So it's difficult to extract from the data, but it's currently a very interesting question. In those patients who have negative testing, are they negative because they don't have genetic kidney disease? Or are they negative because we don't currently have the tools to diagnose testing? And a prime example is MUC1 testing. MUC1 testing is, and I think you're going to be talking about it, is frequently missed on standard genomic platforms, and you need specialized methods in order to diagnose this. So for some patients, when the diagnosis is negative on genetic testing, it's negative because we don't have the tools to diagnose it yet.
So that we're still learning more about different genes, and also there's still limitations in our methodology. And I think also you'll see when you get the results back, sometimes there'll be a variant of uncertain significance. However, if you read the genetic mutation and then their clinical phenotype, it appears like it should be considered pathologic. Is that also a limitation of the amount of knowledge we have about specific gene mutations and how they lead to disease at this point in time?
I think variants of uncertain significance need to be handled appropriately. They are labeled variants of uncertain significance because it is just that. We're not certain whether this mutation is disease causing or not. It is important to remember that some of these variants may be upgraded to a pathogenic classification, but it's also very important to remember that some of these can be downgraded to a benign classification. Again, to use an analogy in radiology, sometimes you see an abnormal finding on a scan, let's say a cyst, and it has some high risk features or low risk features. And often we say, let's repeat the scan in 6 months or a year and we can reevaluate this. It's exactly the same with genetic testing. If you find a variant of uncertain significance, the current suggestions are to repeat the testing or repeat the analysis in 18 to 36 months. And over this time, additional information may come into the system, either from the patient, they may have additional clinical features or different family involvement that can be added into the analysis, or there may be external data in the terms of functional data. If this is added into the analysis, this can help in the reclassification. So I think this is just going to become part and parcel of genetic testing. And we just got to be very cognizant that when you're done genetic testing, maybe you're not done. Maybe you need a reanalysis in 18 to 36 months.
Well, the next figure I wanted to talk about is figure 4, and this describes the downstream actions that occurred after the genetic diagnosis. This could include things like testing family members, changing therapies, and I'm really, really interested to hear about what you found in this one.
Yes. This is becoming increasingly important because many studies to date have focused on diagnostic yield alone, predominantly in research settings. But things need to move on, and we need to answer these very important questions. Is great, I have a diagnosis, but what does that mean for me as a patient, and what does that mean for me as the caring physician? So overall, I will say across the 60 studies, only 7 reported extensive data on clinical utility. But overall, we did find that 18% of those in whom they reported the clinical utility, that there was a direct change in management after confirming a definitive genetic diagnosis through genetic testing. This probably occurred at the highest frequency when there's a reclassification to another subtype of CKD, because ultimately, if you haven't got the right diagnosis, it's very hard to apply targeted treatments for the patients. So let's say, for example, and in fact, this isn't actually captured in our reclassification figure. So take, for example, glomerular disease. Sometimes you can have within subcategory classification. So, for example, there's a high rate of classification from Alports to genetic forms of FSGS and vice versa. So certainly you can see where there's a direct change in management where you will treat someone with Alports entirely different when you confirm a genetic form of FSGS.
One thing that we've sort of transitioned to is we started with single genes and now we're going to panels. We're also finding things like an APOL1, which we haven't talked about extensively here. At what point do we continue on the panel genetic testing of like 400 genes, or do we also consider single gene testing? And particularly, I'm interested because it sounds like our clinical phenotype in a lot of patients is not that good. A lot, 17% get reclassified. Should we continue down this road of panel testing?
This is a really important question and one we really hope to answer with this. However, I will say our data showed that there was no significant difference in the type of testing in terms of your ultimate diagnostic yield. It was very similar whether you did single gene testing versus panel testing versus exome testing. What did seem to impact the testing was your pre-testing diagnosis or your index of suspicion. So let's say those patients who had tubulointerstitial kidney disease that underwent MUC1 and UMOD testing. In fact, the highest diagnostic yield was in that category. I don't think necessarily it's because of the type of testing that was performed, but it's based on your index of suspicion. So I will say if you have a very high index of suspicion of a disease, single gene testing may, in these limited cases, be appropriate. But for patients in whom we can't say, I definitely know the diagnosis, then a more comprehensive approach to testing, be it large gene panels or indeed exome or genome analysis, seems to be the most appropriate test.
Any other points that you'd like to make about this study?
One important point I would like to make, because it's not often a disease entity we think about in adults, is the high reclassification rate observed within the ciliopathy group. Very few of these cases were suspected in adults pre-testing, and in fact, all of them were diagnosed after genetic testing. And it's important to consider because for a number of reasons, we as adult nephrologists aren't considering this diagnosis, nephronophthisis. There might be a multitude of reasons for this. It's because classically, it's described as a childhood onset disease. Or there may be a publication bias towards publishing more severe phenotypes that are associated with neurological manifestations, which aren't necessarily seen in adults. Maybe it's because we can't spell it. Nephronophthisis isn't the easiest to spell. But for whatever reason, we're not suspecting it. And I think this goes back to our education and training in genetic kidney disease, particularly among adult nephrologists. And really, we need to open the education to the various different types of genetic kidney disease that adults may also have, not just children.
So I guess the final point, should genetic testing become standard in nephrology clinics?
Yes, it should. In appropriately selected patients, it definitely should be. There's a number of ways of looking at this. A number of jurisdictions are looking at different models of service that can be provide this service. There's certainly mainstreaming models where we try to get it into all general nephrology clinics. We could look at instituting it in the current services or the current subspecialty clinics we have, such as, let's say, GN clinics might be an appropriate space for this. Or ultimately, we need to consider having kidney genetics clinics. But whatever way we choose to integrate it, there certainly will be patients who miss out on not just a definitive diagnosis, but ultimately the benefits of achieving that diagnosis if we don't successfully integrate it into clinical practice for these patients.
Well, thank you so much for this discussion around genetic testing. And also, congratulations to you and your group for this publication. I'm really excited to see the future of genetic testing, and studies like this are really important so that we all are on the same page and can move forward together. What are your next steps?
One of the main things that we really want to decipher is a lot of the times in all of these studies that we looked at, we were looking in research cohorts or we were looking at prevalent CKD patients. And ultimately, genetic testing came as a last resort when all other diagnostic strategies have failed. One of the key things I think we need to focus on now in nephrology is If we were to institute genetic testing at an early stage in the CKD workup in our incidence kidney disease patients, what is the role of genetic testing there? So, moving the dial and shifting the genetic assessment to earlier in the disease process. I think this is where we're really going to feel the benefit of genetic testing for these patients.
So there's multiple ways this can be
Yeah.
integrated in.
Yeah.
So, well, thank you so much. We appreciate your time and your effort in this area of nephrology.
Thank you so much. Delighted to chat.
The next paper we'll discuss is from Jason, and the title is Systematic Screening of ADTKD MUC1 Pathogenic Variants Through Exome Sequencing. I'm very excited to be joined by Dr. Moran Davila Lavet from the Faculty of Life Sciences at Bar Ilan University in Israel. Welcome to ASN Kidney Translation. Can you introduce yourself?
My name is Moran Davila Levit, and I'm currently at Bar Ilan University in Israel, where I have my independent lab. And we are working on chronic kidney diseases associated with protein trafficking defects. Previously, I did my postdoctoral fellowship with Professor Anna Greca at the Broad Institute of Harvard and MIT, where we identified the mechanism by which MUC1, which is the gene involved in this current paper that we are going to talk about, so we identified its biological mechanism of action, how it induces or causes the associated disease.
Well, MUC1 is a very interesting disease. I've always had a very big fascination with it. What is the challenge? Because it's not typically a gene that we can readily sequence when we send out a panel for kidney disease. So, what is the issue with MUC1? Why is it so hard to figure out any mutation with that gene?
Right, this is true because this is definitely was a huge challenge for the last few decades, where basically when we applied next generation sequencing, you could not really detect the mutation, and the geneticist did not know where the mutation is actually found. The problem with this gene is that it has what is called a region that is called a variable number of tandem repeats. In short, it's VNTR. And this is basically a region that is composed of many repeats, composed of 60 different nucleotides that can repeat themselves something from 20 times to 120 times. So these are basically relatively large repeats, again, 60 nucleotides that result with 20 amino acids that repeat themselves on average 60 times. And this is something that the next generation sequencing that is based on very short reads cannot detect when you have only a single nucleotide insertion into one of these repeats. So this is something that is super challenging, and I will make it even more complicated by saying that the one nucleotide insertion that is seen in something like 90% of the patients is a cytosine insertion into a region of 7 cytosines. So most of the patients would have 8 instead of 7 cytosines in one of these repeats, and it's very variable in which one of the repeat each patient will have it. And therefore, the next generation sequencing, which is based on these very short sequences of about 25 nucleotides, would miss this one nucleotide insertion.
Well, let's talk a little bit about MUC1. What's the function of mucin 1 in the human body, and what happens when you have this mutation, and how do you see a patient and think they might have this genetic mutation?
So, mucin proteins are basically proteins that are found on epithelial cells, and their function is to present some kind of a barrier between the external surrounding to the epithelial cells. As such proteins, their function is basically to protect the cells, the epithelial cells, the epithelial layer. And we have two types of mucins. We have the transmembrane mucins and we have the secreted mucins. In this case, MAC1 is a transmembrane mucin, so it is found on the apical membrane of the epithelial cells, and it's basically a protein that is heavily glycosylated. So with this huge glycosylated tree, it's water molecule, and this what serves as this protection against different bugs to enter the surface, the epithelial cells that basically cover all the organs that are exposed to the external surrounding.
Sounds like it almost acts as like shrubbery or bushes that surround your house.
Yes, yeah, definitely. And it kind of prevents all the things from getting to the inside of our body. So, yeah, so this is mucin 1, and this is why it is important to those epithelial cells.
So, what happens when you have a mutation that's pathogenic?
So, when you have a mutation, and specifically this mutation, what happens is this one nucleotide insertion into this MAC1 gene creates this frame shift in the translation of the protein. So, starting from the place where the mutation is found, you basically now get this new protein, this new set of amino acids that the cell basically did not know, because it's a totally new sequence of amino acids. And what it does eventually is that you get a truncated protein. So eventually you will get an early stop codon to this protein. So it will be truncated. This protein will not even have the transmembrane domain. So it basically cannot be located anymore in the plasma membrane of the epithelial cells. And what it does, what we have found, that it accumulates intracellularly, and it generates this toxic gain of function. So this accumulation is eventually toxic for the cells, and this is what eventually will kill these epithelial cells and will generate this kidney disease eventually, because you lose the tubular epithelial cells that cannot function anymore, and you lose the function of the kidney.
What does that kidney disease look like?
So MAC1 belongs to a group of diseases that are called in general autosomal dominant tubular interstitial kidney disease. And these diseases in general are characterized by an interstitial fibrosis and tubular atrophy. So the disease is found in the tubular cells, in the epithelial cells of the tubules. So you probably won't see a glomerular damage, and usually you won't see any proteinuria, but there are not really specific criteria or symptoms that you would see in those patients, other than in most cases the family history. So because it is usually a genetic disease, you possibility of having this MAC1 kidney disease through this family history.
So it sounds like this might be something that's missed because it doesn't have a lot of specific things that you maybe can tell it apart from other diseases, and sequencing is very important.
Right. And this is why we actually think that this disease is underdiagnosed, because there are no specific diagnostic tools or symptoms that can point to this specific disease. We really do think that it is underdiagnosed, and therefore there is real need for a method, a pipeline that can actually detect it out of generally perform sequencing for patients.
Okay, let's move on to the paper. And just to clarify, you're not an author on this paper, a researcher who's interested in MUC1. This is out of Paris, France, and the laboratory of Dr. Laurent Ménard. What was the methodology that they had to try to refine the sequencing technique to identify these pathogenic mutations?
So basically, until recently, we weren't able to detect the mutation for MAC1 using next generation sequencing. So you could do it with applying different methodologies that are either very expensive or not very with a high throughput nature. About a year ago, there was a study that was published that was basically generating this pipeline that is called VN Typer. What it basically do is to take next generation sequencing, and it used specific algorithms in order to detect statistically this mutation, this frame shift mutation that I've just explained about. So it could statistically detect these frame shift mutations using this next generation sequencing. So this is amazing by itself. The challenge is that this study that was published by the group of Guillaume Dorval basically used enriched or targeted next generation sequencing data in order to apply this analysis pipeline. That means that in order to do it with the generally used exome sequencing or whole genome sequencing, which has a lot more sequences, it would be much harder because the coverage of MAC1 is lower, and there is basically a whole sea of sequences to kind of fish this MAC1 and detect this frame shift mutation within this entire sea of sequences. So basically, what this current paper from the group of Lorraine Menard did is to integrate another tool that was previously developed that is called Shark, that can retrieve specific regions to fish out the sequences that we are interested in, as MAC1 in this case. So what the current group did is basically to combine these two tools or pipelines of this Shark tool that retrieves desired sequences or regions, and the VN typer that can basically detect out of these retrieved sequences the frame shift mutation. So they basically combined it, and they did some other stuff that I won't get into, in order to make it possible to basically take these exome sequences of different participants and then to detect the frame shift mutation of MAC1, which is pretty amazing.
Okay, so then the shark approach was able to then take out and maybe locate the actual mutation quicker?
So now the idea was whether we can apply this VN typer pipeline to exome sequences, which consist a lot more different sequences. So the authors of the current paper we are discussing, when they basically apply the VN typer approach or pipeline to regular exome sequencing, it took them 6 to 12 hours per a single participant to look for the mutation. So this is a really long time.
Does not sound feasible at all.
Exactly. And yeah, it's not doable, right? When you would like to look at a whole population or an entire cohort of exome sequences. And therefore, even though this approach, it worked and it was very sensitive and it detected the mutation, however, the time it took is not really feasible for large cohorts. And therefore, the application of the Shark tool, which can then take exome sequences and fish out only the desired regions, in this case, the MAC1 gene, made it much more feasible. And then the authors of this paper were basically, they managed to detect the MAC1 mutation in only 7.5 minutes per individual. So this is pretty amazing. And then this really makes this detection of the MAC1 frameshift mutation feasible and doable for large cohorts.
Wow, that's huge. So from 10 hours to less than 10 minutes. Now we have something that's potentially feasible that we can screen on more people. So that's great. How Yeah, how did they first test it on a cohort of patients?
So they basically started from a proof of concept cohort where they took around 300 individuals where they knew exactly who of the participants is positive for the mutation. So they had their positive cohorts and the participants that they knew that they are negative, and they tested their new combined pipelines, and out of the 33 positive individuals, they could basically detect 32. So one was missing. And the other negative cohort, which they had 54 negative, known negative individuals, all were indeed negative, also using this Shark VN typer approach.
So just, I mean, it sounds like it's pretty good, but why was one unable to be tested positive that was positive for the original technique?
Not clear. I think that the authors suggest that maybe I didn't mention in the beginning, but the MAC1 gene, other than this repeated region, the repeats themselves also they have some variations. So I think that the authors suggest that it might be a different type or variation in the repeated regions that made them miss this one case.
Let's move on to the retrospective cohort,
Right.
and they screened for 3,512 participants that they already had exome sequencing on.
Right.
And what did they find there?
So there they found that 23 participants were positive for the MAC1 mutation. And they took the DNA samples for then a follow up snapshot PCR test that can actually confirm the presence of the mutation. And in this case, 5 individuals out of the 23 were actually negative. So out of the 23 that they detected the mutation, 5 were negative. So, but 18 indeed confirmed to be positive for the MAC1 mutation.
And I think that's, to me, I guess the scary thing is that there are likely many people who have had genetic testing who have
Mhm.
MUC1 who have no idea they have it.
Right, that's true. That's true. And I think this is why I think that this is really an amazing tool or pipeline that could now would make diagnostic much easier than before. And patients that either do not know of the existence of MAC1 or the possibility that there are also de novo cases for this mutation could be now diagnosed with this pipeline. So this is pretty amazing.
And then the last step was a prospective cohort of 825 patients. And what did they find there?
Right. So out of these about 800 patients, they detected 13 that had the MAC1 mutation. And again, after they performed this downstream snapshot PCR test that can validate and confirm, one patient was negative. So 12 out of the 13 were indeed positive for the MAC1 C insertion. So this is a 12 out of 13. This is pretty good. And again, remember that these are exome sequencing data. So it's pretty amazing that it is doable in a relatively short time. This is really great.
Obviously, this is pretty early in moving this forward to actually be able to make it to patient care. Do you think this is a technique that is going to allow us to use in patients all over the world? Or do we still have more refinement to go?
I do think that we should definitely advocate for it and to integrate it into the general routine. But as any other bioinformatic tool or pipeline, definitely I think that the more you do it, the better it gets. So definitely there will be more refinements and there will be more that is done. The other thing is, for example, another consideration is that as I mentioned, most patients do have this cytosine insertion generating the frame shifting MAC1 translation. However, there are more patients that have different mutations. We don't know yet whether this pipeline will be able to detect or identify the other mutations, for example. So this will be something that will be very interesting and important to test as we go with this application of this pipeline.
The last comment to make, it seems like MUC1 and this mutation in the future might have potential therapy. Now, that's obviously still in developmental stages, but it's really important to identify patients that might need to be enrolled in clinical trials to test those therapies out. And right now, we just don't have an ability to do that. So, what does this do for your research and others that are interested in MUC1? Can you talk about that?
So, yeah, definitely. So, yes, as I mentioned, my study together with the group of Professor Anna Greca at the Broad Institute, we have identified the mechanism responsible for this disease, but we also identified a very promising drug or compound that is currently under development. And this is why I think these kind of tools to identify new patients will be super important for the future clinical tests that we will hopefully get into doing with this promising compound, and also for future development or looking for a different, we are still looking for different ways and different strategies to tackle this issue and to find a therapy for this really toxic protein. So this is a really important step for helping us in the future to combat this disease.
Well, thank you so much for being on the ASN Kidney Translation podcast. It's really exciting to see advances, especially integrating genetics with potential therapeutics is the future of our field. And so thank you so much for taking time and sharing your wisdom with us.
Thank you very much, Matt. And I really would like to thank the entire scientific community that is really seeing the importance of working on these rare genetic diseases, and especially MAC1, which is close to my heart. So this is amazing, and it's great to see the development.
The final paper we'll be covering is in Kidney 360, titled Clinical, Pathological, and Genetic Characteristics of Patients with Digenic Alport Syndrome. We have Dr. Michelle Rowe, who is a pediatric nephrologist and division director of the Pediatric Nephrology Division at the University of Minnesota. Welcome to ASN Kidney Translation. Thanks for joining us today.
Thanks for having me, Matt. I'm always happy to talk about Alport syndrome.
Every time I see Alport syndrome, I think of you. And this is a paper that I don't read a lot of papers on Alport syndrome. So I thought I'd bring in the expert to sort of help us and guide us through this paper. Before we do that, can you tell the listeners a little bit about Alport syndrome and the array of different genetics that you can see in that syndrome?
Alport syndrome has always been a disorder with complicated genetics. There are X-linked forms, autosomal recessive, autosomal dominant, and the more we learn about Alport syndrome, the more complicated it gets. So the most common form of Alport syndrome is X-linked Alport syndrome, and that's caused by variants in COL4A5 on the X chromosome. So of course, males are more affected, females can be less affected, but can also have end-stage kidney disease. Then there are autosomal variants in COL4A3 or COL4A4, and those, if heterozygous, cause autosomal dominant disease. And if they have two variants in COL4A3 or two variants in COL4A4, they have autosomal recessive disease, which acts very much like an X-linked male.
Why don't we pause right there? And I think this is something that you have taught me earlier in my career, and I've continued to try to teach our fellows and others. Can you talk about the X-link in the female and removing the terminology of carrier in that situation?
Yeah, I've really tried to push people away from using that term carrier for women with X-linked Alport syndrome. And that's because women with X-linked Alport syndrome don't just have a hidden gene, that may cause disease in a future person. They actually manifest the disease. So almost 100% of women with X-linked Alport syndrome have at least hematuria. The majority have proteinuria at some point in their life. And up to 20 to 30% may develop end stage kidney disease. So having the label of carrier really makes providers forget about the risk that those women may be at to develop progressive disease, and they tend not to treat them as aggressively as you might a male with disease, but it may be just as important to treat those women with X-linked Alport syndrome.
Okay, so that's X-linked. So what about all of the other mutations that can be seen causing autosomal dominant Alport syndrome?
So, autosomal dominant Alport syndrome is becoming a really hot topic because this is coming up more and more in patients who have genetic testing done for FSGS or even for classic Alport syndrome. We're seeing those heterozygous variants in COL4A3 and COL4A4 popping up more often. And there was a study that came out a couple years ago looking at the frequency of those variants just in the general population. And they found pathogenic COL4A3 or COL4A4 variants in 1 in 106 people. So that's a huge frequency. So it tells us that if you go looking for these variants, you're going to find them. But what we really don't understand yet is why do some people with heterozygous COL4A3 or COL4A4 variants go on to develop disease, and others have no phenotype whatsoever. So I think that's really the cutting edge of what we're trying to learn about the genetics of Alport syndrome.
So just to summarize for our listeners, so collagen 4 is what is mutated in Alport syndrome. The A5 is on the X chromosome. A3 and A4 are not on the X chromosome and are considered autosomal dominant. Is that correct?
That is correct. And each of those gene products, they make a isoform of type 4 collagen, and those three isoforms get together and form a trimer. So all three of those gene products are necessary to make up the normal glomerular filtration barrier. So that's why absence or mutations in any one of those three causes disease.
And before we get to the paper, I just want to just verify one thing. As you mentioned, the pathology, and we classically think about alterations in the glomerular basement membrane leading to this basket weaving, and then you alluded that autosomal dominant mutations in 3 and 4 and for Alport syndrome can have a much variable presentation. Can you reiterate that?
It's probably been 10 years now that we've known about the association between FSGS on biopsy and type 4 collagen variants. And it's often the COL4A3 or COL4A4 variants that are popping up for those. Clinically, these patients can look just like a primary FSGS patient. They have nephrotic syndrome, they have a lot of proteinuria, their biopsy on light microscopy shows focal sclerosis. And then when you look at their basement membranes, some of them have classic basement membrane findings of Alport syndrome. Some just have thinning of their basement membranes, but others have really quite normal appearing basement membranes. It's unclear whether these variants are then the primary cause of the patient's FSGS or if it's just a risk factor that's making their FSGS worse. We have a lot to learn about the pathogenesis of that.
You mentioned earlier that the X-linked or the mutation in A5 was the most common. Is that because we see these patients come to clinical care, and now that we're doing more genetic testing, is that kind of flipping and we're seeing more of these 3 and A4 mutations?
Yes, we're definitely seeing more of this as we're doing more panel testing. Even 20 years ago, when I first started seeing patients clinically, if we thought it was an X-linked disease, we would only get the COL4A5 genetic test because it was so expensive to get all three of the genes. Now, you almost always get every single, every gene under the sun in your panel and can get a little bit more understanding of the nuance. So people have been surprised in families where they thought it really looked like X-linked. They're doing the full panel genetic testing, and maybe it's autosomal dominant instead. So you never exactly know what you're going to get by examining pedigrees. Our families are so small these days, for the most part, that you're not going to see the classic pedigrees that you see in the textbooks.
I think that's a good segue to what we're talking about in this paper, and that's digenic mutations. And so, can you tell us what does that mean?
Yeah, so digenic variance is simply when the inheritance of two genes leads to a phenotype that would be unexpected with just one or the other. And the phenotype can't be explained by the individual gene defects, but it's based on their functional interaction. So, for example, in Alport syndrome, a digenic variant might be if there's a variant in COL4A3 and a variant in COL4A4, rather than two variants in COL4A3, for example. Or if a male has a variant in COL4A5, but they also have a variant in COL4A3 somewhere. So it's the interaction of those two different Alport syndrome variants and how they cause disease. One of the first studies that showed digenic inheritance in Alport syndrome was published in 2015 by Alessandro Ranieri in the Journal of Medical Genetics. And they found a number of these patients that were out there. And it was really the first time that we started to think about how digenic inheritance might behave differently than patients with classic Alport syndrome.
One question is, how often do these digenic variants occur?
In this study that we're talking about today, they found digenic inheritance in 1.7% of the patients that they did screening on. And this was about over 800 patients over about 20 years of analysis. So they don't come up very often. This is similar to findings from the Athena study. And this is an unpublished study that was done in the US. This was a natural history study of patients with Alport syndrome. 165 patients were enrolled, and 2 patients out of those had digenic inheritance, but it's not super common. And that's why we don't have very much information about it.
What was the question that the researchers were attempting to answer?
So this was a study from Kandai Nozu in Japan, and he was really looking at what is the difference in phenotype in patients with digenic disease versus those with more classic Alport syndrome. So if you are a male with X-linked Alport syndrome, for example, is it worse for you if you also have a COL4A3 variant, or is it worse if you also have a COL4A4 variant? So those were the questions that they were trying to get at.
Just trying to think about it. It sounds like the hypothesis would make sense, because if you have 2 mutations, would be worse than just having one.
Absolutely. Yeah, more mutations is worse.
But really, it hadn't been systematically studied in a large group of patients to really show that.
Correct. Yep. And I think another idea is that, well, if you already have one variant, what does it matter if you have another one? Their disease is already as bad as it's going to get. So it doesn't really matter how many variants you have.
Well, what were the methods that they used to try to answer this question?
Yeah, so this was a relatively simple retrospective observational studies. So they took patients with suspected Alport syndrome who were referred to their center, and they did genetic testing. And then they went back at their clinical outcomes. So when proteinuria and stage kidney disease, findings that were associated with the patients who had digenic disease. They found a total of 27 patients and 14 families. So there were a couple families who had more than one patient that was included.
Okay, so what did they find?
They took patients and put them into a couple different buckets. The first bucket were patients who had one variant in COL4A3 and one variant in COL4A4. So they had these two different variants. And they looked at the age at detection of proteinuria. And for patients with digenic inheritance, they develop proteinuria at age 10 compared to age 20 for patients who had autosomal dominant disease. And they also looked at the age at end stage kidney disease, and that was also earlier for patients with digenic inheritance. They reached end stage at 57 years compared to 72 years for autosomal dominant. There were similar findings when you looked at male patients with COL4A5 variants who also had a COL4A3 or COL4A4. They developed proteinuria quite early at age 5, and they also developed end-stage kidney disease at 27 years, which was earlier than the other cohort.
That's a pretty stunning finding if you look at the ESRD 70 to 57. So almost a 15-year difference.
Correct. And these are small numbers, but it is a real difference. This is the largest study that we've really had on digenic disease. So I think it is telling us, especially in these patients with the heterozygous COL4A3 and COL4A4 digenic inheritance, that this is potentially important information.
One of the things I noticed in Table 3, it listed the pathogenic mutation as being likely pathogenic for almost 80 to 90% of the mutations that they identified. And as someone who's doing some genetic testing now, I'm getting this back. Can you tell us about how to interpret that?
Sure. So these characterizations come from the American College of Medical Genetics and Genomics, or ACMG. You'll hear people talk about ACMG criteria for classifying variants. And pathogenic means that we have ample scientific evidence. We know for sure that if you have this variant, you're going to have disease. So the likely pathogenic variant leaves a little bit of wiggle room where you expect that greater than 90% of those variants will someday go on to become pathogenic. So you're pretty sure that a likely pathogenic variant really is the cause of disease, but you can't say with 100% certainty. It allows that uncertainty to be there. And then one level below that is that variant of unknown significance, which none of us like getting back on our genetic testing reports.
So likely pathogenic would still be classified as pathogenic in those reports.
Yeah, most reports that come back will combine pathogenic and likely pathogenic because we think about those clinically in the same way. Those are probably what we're looking for, and those are probably the cause of disease.
What are the limitations to this study?
The main limitation is really just the small numbers that they had. It's very hard to put a big group of these patients together, and I could imagine combining a number of registries that are out there in Alport syndrome to try to get bigger numbers together to really understand what do these variants mean for people.
What are the implications that these findings have for patients with Alport syndrome?
If you find digenic inheritance, it can help you as their physician really counsel them about the risk more effectively. So if they have that digenic inheritance, you may be more aggressive about treating them with ACE inhibitors or SGLT2 inhibitors, even if they have just the COL43 and COL44 variants. It also may be able to help explain disease in a family that seems out of place. So, if you have a family with X-linked Alport syndrome, for example, who have always developed end-stage kidney disease at right around age 30, and now all of a sudden you have somebody in the family who's reaching end stage at 20, maybe we need to re-examine their genetics to see if maybe now they have a COL43 variant to go along with their COL45. So it can help a little bit with all of that nuance of phenotype.
It's sort of a dream because I remember starting in fellowship, doing genetic testing was just not something that was feasible. And I think as a community, it's exciting to see us actually being able to say, here's why you have kidney disease. And to me, that is really rewarding. And it could lead then to specific therapies down the road. Can you comment about how genetics has changed and where you see it going?
Yeah, absolutely. I agree. Genetic testing is so much more available, and it's so valuable for patients to have a name to their disorder and to know that this is why I have it and this is what's going to happen to me. I think genetic testing is really important. And that name is also something that is changing. So as we're doing all of this genetic testing, we're finding people with more and more of these variants in type 4 collagen, and we don't have a good nomenclature for what to call those patients. It's not quite right to call them all Alport syndrome because some of them may never have any phenotype. They may just maybe they just have microscopic hematuria. So there's a group that is putting together naming recommendations that should be out shortly. They're starting to circulate some draft suggestions for what to name these patients. So I think that will be a nice first step in moving forward with how we use these genetic results.
Nomenclature and naming has a lot of implications. And I was just curious in that regard, like you have the Alport Syndrome Foundation, and if it does change, the name changes, and you have these individuals that maybe don't have Alport syndrome, they have a collagen mutation, who's going to help advocate for them and to get therapies? And how do you see that changing in the future?
Yeah, and that's a really important question. And I know a number of the patients that I talk to, not all of them, but many of them really do connect with the Alport name, with that it is important for them to retain that Alport syndrome name within the name of this spectrum of disorders. So I suspect that Alport will be retained in there somewhere, but I'm not sure what the final product is going to look like.
We have a lot to learn, and it's exciting time to be in nephrology. Thank you so much for taking time out of your busy schedule to be with us, and we look forward to seeing therapies and new discoveries in Alport syndrome in the future.
Absolutely. Thanks for having me.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.